it's Hannah Johnson from the BIA here, welcoming you to the BIA webinar on advances in cancer research. I'm delighted to have two of our members online today, talking their company updates. Firstly, we have Dr. Richard Goodfellow, Co-Chief Executive Officer of Scancell, and also we have Jeff Munkle, General Manager of Champions Oncology. Just a few things before we start. If everyone who's not a speaker can mute themselves, just to prevent any background noise. And also we'll be waiting to the end to do all questions. If you've got any questions, you can either drop them to me on the chat on the bottom right-hand screen, or I'm muting everyone at the end so you can say it at the end to us. Okay, so without any further ado, I'm going to pass over to Richard Goodfellow to talk about Scancell.
Me. Hello?
We can hear you, yeah.
Yeah, good. Okay. Well, good morning, everyone. I want to talk to you in particular about the exciting future for cancer vaccines. And I think it's fair to say that until recently, there's been a certain amount of skepticism about the viability of cancer vaccines. There have been a number of high-profile failures, and even the successes, Provenge and T-VEC in particular, have suffered from rather weak efficacy data, you could say, not to mention issues with manufacturing and delivery to patients. T-VEC is intratumoral, for example. But that perception is changing. And as new, more powerful technologies emerge, for example, only a couple of days ago, Rintega from Celldex, which is a vaccine for glioblastoma, reported really quite dramatic increases in overall survival in an incredibly hard-to-treat cancer. And Scancell SCIB1 is also showing really quite exceptional survival benefits in stage 3-4 patients with melanoma that's been resected. And a further boost to the potential of cancer vaccines has been the realization that they could probably increase the response rate to checkpoint inhibitors by enhancing the innate T-cell response. I think we all know that checkpoint inhibitors have been a fantastic advance, but they only work in 25 to 40% of patients, and at the moment it's not possible to tell which ones. So if you could add something to that regime that would increase response rate, that would be very desirable, and it's thought that maybe cancer vaccines can play that role. And then finally, you've got the issues of safety and cost. If you want to combine checkpoints or anything else with another agent, it's better that that agent is well tolerated. And cancer vaccines, for the most part, are much safer than other forms of cancer treatment, and that's certainly the case for Scancell's immunobody vaccines. So let's move on. There's our disclaimer as a public company. So we set out on this story with the belief that if you want to have an impact on cancer through a T-cell response, you need to generate ultra-high avidity T-cells. It's no good having plenty of T-cells if they're not functionally effective. So right from the start, the screening for the best candidate vaccines was based on which constructs would generate the highest T-cell avidity. And with regard to our lead platform, Immunobody, we discovered that the combination of the Immunobody approach, which I'll describe in a minute, delivered as a DNA plasmid with electroporation, did the best job. And it generated potent killer T-cells with the avidity at least equivalent to cell therapy approaches, but in a much simpler way. And that is indeed the basis for our SCIB1 melanoma vaccine that's now in the clinic. So today I haven't got much time. I'm only going to talk about Immunobody. That generates, by and large, CD8 T-cell responses. It's a DNA product. We think it's great and can be great in combination with checkpoint inhibitors, but very effective in eradicating small primary and metastatic tumors as monotherapy. and it takes T-cell epitopes from target antigens and uses the immunobody construct to deliver them very effectively to the immune system. Moditope is our new platform. It's a peptide. In fact, it's a series of citrullonated peptides that generate potent CD4 T-cell responses. And at least in animals, that's reversing the immunosuppressive tumor environment like checkpoint inhibitors. So there seems to be no need for checkpoint inhibitors, and we hope it will be perhaps an alternative to checkpoint inhibitors in the future. So that's what we're doing. I want to talk very briefly about the clinical results we've got with SCIB1 because it's very promising and we think confirms and validates the viability of our approach. So SCIB1 has been in a phase 1/2 clinical trial for a number of years. Part 1 was in patients largely with inoperable tumors. Part 2 was an extension phase in really fully resected patients. So these are the patients we think we will have the most benefit from. You can see there in that little diagram on the right-hand side what an immunobody is. It's actually a humanized antibody into which T-cell epitopes have been engineered into the CDR regions. And that antibody then acts as a vector for delivering those epitopes via the Fc receptor at the bottom to dendritic cells. So it's a delivery system. And our objective from this study was, of course, to show that it was safe, well tolerated, to find out the maximally tolerated dose, and to look at immune response and to a certain extent efficacy, and that's what we've done. So the study was of 6 months duration, 5 vaccinations, immune samples taken pre and post dosing, and during the course of the study we were given approval by the regulatory authorities to treat for up to 5 years. And some of our patients have now been treated for over 4 years and are doing very well. If you don't know about electroporation, this is what it is. It's the use of an electric current at the same time as you give the intramuscular injection. And the idea is to generate transient porosity in the cell to get very high concentrations of DNA inside the cells that you're targeting, which you could not otherwise do without electroporation. We do this through our collaborator in the States, iCore Medical Systems, and the commercial-ready device is now being manufactured by Flextronics. So any future trials will be done with a commercial-ready electroporation device. That's electroporation. The first thing to say is that we get a really excellent safety profile from SCIB1. If you compare it with just about anything else that's being used in melanoma, it's very well tolerated. So there were no adverse events leading to discontinuation of treatment. The most common adverse events were related to the electroporation itself, which is largely transient. There is some pain in some patients, but they, given the benefits that we hope will accrue from treatment, they come back and are happy to have further injections. We've given it now around 200 times, and we see no reason why this cannot be given very safely and widely in the future. Just some highlights from the trial. In part 1, which if you remember was in patients with inoperable disease, we saw at quite an early stage in the study some patients with metastatic tumors in the lungs. We saw a dramatic reduction in those metastatic tumors or even elimination. So that's a very rare spontaneous event, as you all know. So we knew at this stage that we had active treatment. Virtually all patients had a melanoma-specific immune response. So we know that it was doing what it was supposed to do and did do in animals. The only patients who didn't respond were those that were on a very low dose. And importantly, over the patients who've been treated now for several years, that immune response is durable. When we revaccinate, we get the same or even increased immune response. And very importantly, those patients that we are targeting primarily for a cancer vaccine, these are patients with already a strong immune system. all 20 patients with resected disease are still alive, and that's pretty remarkable. This is just a very brief summary of the survival data. We have 16 patients treated at 2 to 4 milligrams of SCIB1, and they've been treated, most of them, apart from 2, for at least 3 years. And if you look at the left-hand columns, you can see they're all still alive, but importantly, 70% of them, regardless of whether they had stage 3 or stage 4 disease, have no signs of recurrence. If you compare that, for example, with Yervoy, looking to the right, you'll see that after 3 years, we don't have the overall survival data, but the recurrence-free survival data in the placebo group was 35%, which only increases to 47% in the treated group. So we would agree that our study is small. It's also open, but the data is looking increasingly compelling. We have another 4 patients at a higher dose, at 8 milligrams. Those patients haven't been treated for so long, but they all are alive, and none of them have any disease recurrence at this stage. So very exciting results. The only other treatment for adjuvant therapy in the melanoma field that has approval at this stage is Yervoy. It was approved a very short while ago. Having spoken to melanoma specialists, very few of them will intend to use Yervoy in resected patients, and the reason is it's horribly toxic. You can see that there was a quite modest benefit for Yervoy patients, 46 versus 34%. That compares, as you all know, with 67% on SCIB1. But in the Yervoy group, 52% of patients discontinued treatment due to adverse events, some of them very serious adverse events, and 5 of those patients died due to drug-related adverse events. So if you're considering using Yervoy as an adjuvant therapy, it's really quite a tough call given the potential for causing harm. Finally, I just want to say something about the rationale for combination. We very much like the idea of using cancer vaccines for adjuvant disease, but we all recognize, I think, those of us working in this field, that checkpoint inhibitors will dominate the treatment of later stage disease for some years to come. As I said, for early tumors, vaccines play an important role. We think for late tumors, checkpoint inhibitors will obviously play an important role. And we think combining checkpoint inhibitors and vaccines makes a great deal of sense, because checkpoint inhibitors will expose the tumor to attack by T cells, and the cancer vaccines like SCIB1 will provide a more powerful T cell response, which should enhance response rates. And certainly in animals, we've done a lot of experiments to show that that's indeed the case. The theory translates into practice, that if you use either SCIB1 or a PD-1 drug alone, you get a survival benefit compared to no treatment. But if you combine the two, you get a much more potent survival and anti-tumor benefit. So as well as monotherapy, we see a combination therapy as an exciting opportunity. So just to close, I think I was told I had about 10 minutes. This is our vision for the treatment of melanoma by stage. If you have early stage disease and a tumor that has not penetrated to beyond a few millimeters depth, surgery does the job. If you have late stage disease, stage 4 disease, checkpoint inhibitors and other more potent agents are probably required. If we go back to stage 3, we think, and perhaps late stage 2, the combination of checkpoint inhibitors and SCIB1 looks to be an attractive combination. But if you go back to stage 2 and early stage 3 disease, which is very common, very large numbers of patients, then in the adjunctive setting, agents like SCIB1, which are very safe and seem to be effective, offer a great chance of treating patients where, at the present time, there are no alternatives. So, Hannah, hopefully, and Rachel, hopefully I've stuck to my timing, and that concludes the presentation.
Great, thanks very much, Richard. We'll now be passing over to Jeff Muckle from Champions Oncology. So over to you, Jeff.
Thanks, Hannah. Can you guys hear me?
Yes, we can.
Fantastic. Cool. Just firstly, Richard, thanks for kicking that off. I think that SCIB1 and Scancell have certainly demonstrated how we are advancing cancer research, certainly in immunotherapy space. So that's pretty exciting. So essentially, Champions Oncology are a PDX company. What we do is we work with oncologists and private patients, whereby we take cancer tumors from patients and we grow patient-derived xenografts, or we call them tumor graft models. And the purpose of doing this really is to develop these mice models in order to conduct drug sensitivity testing. Now, for those of you on the call who have worked with PDXs in the past, will know that these patient-derived xenografts very, very accurately recapitulate human biology. And it's because of the way that they're engrafted. Those scientists of you out there will know that historically cell line derived xenografts where you suspend single cells or neoplastic cells or cancer cells, you can grow those tumors in mice, right? But very often they don't really represent the patient's tumor very accurately because you've interfered with the microenvironment, you've taken out those single cells, and you've grown a tumor. So what we do with patient-derived xenografts is essentially we take a chunk of tumor from a patient, so including the entire microenvironment of that tumor, and we implant that into the mice. And studies, I mean, there are many publications now, and we've published on this as well, showing that by doing this, you retain genetic expression, you maintain antigen expression, and even the histological or the pathological microarchitecture is retained. So I think the debate around, patient-derived xenografts has been had over the years and has now been shown to be an extremely accurate model, not only for the research of cancer, but also for preclinical drug development and clinical drug development. These patient-derived xenograft models are now being used pretty effectively. So what we do from a process point of view is we take a piece of tumor from a patient, we grow it in a first generation of mice. It generally takes between 6 weeks to 4 months to develop these tumors in these mice before we then implant them into a second generation of mice. And at that point, we then are able to do drug sensitivity testing. So whether that's for individual patients in clinical centers around the UK, where we can test standard of care agents, we can test investigational agents, we can test non-conventional agents in these mice. But these mice have been shown to be over 90% accurate in predicting patient response. And I've got some data a little bit later I can show you. So Champions Oncology are a global company. We've got implantation centers around the world. We've got about 5 implantation centers in the US. We've got one here in the UK. We've got one in Israel. We've got one in Singapore. And we have a number of physician or clinical collaborations where we are referred a number of patients to grow specific tumor models for. And then what we do is we have consent to use these models with collaborations with biopharma. Those individuals out there who are developing cancer drugs, we then are able to utilize these very clinically relevant models to test therapies. So I'm just having a look to see where we are in the presentation or what slides we have. Okay, fine. So I'm happy now to, as a bit of an introduction, happy to move on. So as explained, Champions Oncology have over 1,000 models, and all of these models come highly characterized. So you can see that in this slide, we have the tumor profile, well characterized. We have the patient's medical history as well. So for a specific PDX model, we have the treatment information of that patient and the response data and the duration data of that patient. Obviously, we have the patient demographics, but then we also do whole exome DNA and RNA-seq analysis on these models. So if you're a drug developer and you're looking for a specific genetic mutation that you're targeting, it may be P10, it may be KRAS, HER2, maybe PDL1, then you would be able to search our database. And on this slide, you can see that we give full access to anyone to our database, where you'd be able to go in and search our models, and you'd be able to look at patient demographics. You'd be able to look at pre and post collection treatment. And this is just a little example of the ovarian cancer models that we have. We have just over 30 platinum pre-treated models, and you can see that these models have all been pre-treated with platinum, and you can see that most of them have been treated with taxanes and so on and so forth. So this is just a slide showing the relative responses to carboplatin paclitaxel and doxorubicin as well for these specific models. This is just an example in ovarian. We have a number of models across all solid tumor areas. So talking a little bit about our operations. So Champions Oncology headquarters is in Hackensack in the US. And we have a med affairs we have a med affairs team that work globally. We currently have around 85 FTEs, most of them with PhDs or MBAs. Our facilities are pretty good in the US. We house around 5,000 cages, and I think we can take up to around 30,000 mice. Our vivarium and headquarters have all the necessary accreditations as you would expect, and as I've already explained, you can see the implantation centers around the globe there. So just really coming back to what Champions can offer really for individuals who are developing cancer therapies is we have a medical oncologist, which is pretty rare given some CROs. In fact, our medical oncologist developed Tarceva and brought that to market. We have recognized industry R&D. We have bioinformatics specialists, a regionally based team, and of course a fantastic lab with a really experienced lab director there. I've already explained this. We give our data to all customers, including tissue microarrays and FFPE samples for our models. This is just a little flow diagram showing our study process. So if you're an oncology drug developer and you came to us to test your investigational agents in our models, we would run a needs assessment, and we would look at our models. We would then design a clinical study for you, and we would then hand it over to a dedicated project leader who would then actually conduct the study and provide you with weekly updates, etc., etc. So patient-derived xenografts, really, this is an interesting little publication from EMD Sirona showing, you know, the cost differences between your typical phase 2 study cost per patient and PDX study costs. And generally speaking, PDX works out to be more than 10 times cost effective. Obviously, that makes sense. But more specifically, you're able to make, you know, your go no-go decisions a lot earlier, and these, let's not forget, these PDX models are extremely accurate and can predict your success in those molecules. So the next two slides might be quite interesting in that these are the typical kind of studies that we run. If you look on the left-hand side of the slide, you'll see there, you know, through these PDX models, we are able to run typical proof of concept or mechanism of action type studies. We can run efficacy or combination studies with your investigational agent. And looking at the next slide, you know, we could do indication screens, pipeline screens. We could even support in biomarker discovery, clinical trial simulation, and now what we're doing is co-clinical studies as well. So if you're running a phase 2 or phase 3 clinical study, what we do is we develop a PDX for all patients who are currently enrolled in the study. And the reason why you can develop these models is so that any hypotheses generated from the study itself, you're then able to test in the mice, whether it's mechanisms of resistance, whether it's further combinations, whether it's testing your investigational agent with different agents or standard of care. You're then able to use the PDX models to run different scenarios. So that's looking pretty good. And Richard, you'd be pretty interested to know that you know, we now have our immunograft, whereby we humanize. We can successfully humanize mice. So we generally use NOG or NSG mice, which are severely immunocompromised. We then reconstitute a human immune system in these mice. Generally, we do that by reconstituting with CD34 hematopoietic stem cells. And then essentially we wait between 10 to 12 weeks before we see, you know, appropriate levels of CD45 T cell circulation. At that stage, we are then able to implant the tumor type that it may be melanoma, it may be breast, GBM, etc. We then implant those tumors, and then we're able to screen against immuno-oncology agents. You know, we look at tumor growth inhibition. You can look at proliferation or activation of T cells or infiltration of you know, CD4, CD8, and this is extremely exciting. We've run a number of studies with various immuno checkpoints and even with some vaccines as well, Richard, which is pretty exciting. But these models are extremely accurate and are predicting success. So essentially, I just want to conclude by saying that through patient-derived xenografts, we are really making huge advances in cancer research and drug discovery as well. And we're using these models now across discovery, preclinical, you know, late phase clinical development, and obviously translational research as well. And we believe that these PDXs are really the future of research and drug discovery, because they very, very, very closely and very accurately recapitulate human tumor. And that's me.
Great, thanks, Jeff. We're just going to unmute everyone now and put out for questions. So if anyone's got any questions, please say so now. I've got a few on the chat that I will be reading out as well. Okay, so one of the ones on the chat is, so this is for Richard, bearing in mind Scancel's collaboration with ImmuneID, how significant an impact would the ability to predict patients' immune responses have on future trials and value of SCIB1?
Well, it's part of our continuing efforts to understand what is going on. We announced our collaboration with ImmuneID a couple of months ago. The work is ongoing, and all being well, we'll have something more to say by the end of this year. So it all depends on the results. We'll see what those results are, and hopefully it will add to the body of knowledge that will allow us to choose more effective ways of treating these patients.
Thanks, Richard. There's also another one here for you. So, are there any plans to clinically trial the combination of SCIB1 with checkpoint inhibitors? And if so, do you think that they will be eligible for accelerated approval?
Well, as I said, one of the studies that we think should be done is a combination with one or more checkpoint inhibitors. Whether we do it or whether a partner does it remains to be seen. I think it's highly likely that such a study will be done because I think there are animal studies for increasing the response rate from 30% to 50%, which will be very important. Those plans are in place or being put in place.
Great, thanks, Richard. Anyone got any questions from the participants at the moment? I've got another one on chat I could ask. No? Okay. So Richard, there's another one for you. Can immunobody and metotope be combined to have a greater impact, or do they work in isolation?
We think they should be in different groups of patients, as I indicated. So, I mean, bodytope is preclinical, so it's really very hard to draw any particular conclusions about that at present. The immunobody technology we think is particularly suited for earlier stage disease where you have micrometastases or later stage disease in combination with the checkpoint inhibitors. Whether or not immunobody could be combined with Moditope, I think it's really too early to say. The first thing we need to do with Moditope is select our target initial indications and see if it does what we hope it will do in patients, as we've seen in animals. So a bit too early to say yet, but they're on rather separate tracks at the moment.
Okay, a final one for you, Richard. Could immunobody and Moditope be used in targets outside cancer in other chronic illnesses? No. I mean, we are entirely focused on Moditope. There may well be uses of citrullinated epitopes. I mean, they're known to be of potential use in autoimmune disease. We have not investigated that, and our patent position does not focus on that. Our company is primarily interested in the utility of these treatments for cancer. And whilst there are theoretical possibilities of using them outside cancer, that's not our level of expertise, and we have no plans to investigate that at the moment. Great. Thanks, Richard. Final, any questions, anyone? Otherwise, I'm going to move on.
Yes. Could I ask a question to Richard Goodfellow?
Yes.
Is there any way that the SCIB1 trial could be expanded to try to further evaluate the effectiveness of the vaccine?
Well, we're not planning to expand the trial. I mean, recruitment to the trial is now closed, as you probably know. Patients are being followed up for as long as they need treatment. And as you know, all 20 patients with resected disease are still alive, and they will be followed up as long as the regulatory as long as we're allowed to do so by the regulatory authorities, which is a total of 5.5 years. The next step for us, that really is an obligation to those patients to continue. The next step for us, or indeed a partner, would be to conduct new studies, both in combination with a checkpoint inhibitor and more ambitiously as monotherapy for the treatment of resected melanoma, with the goal of preventing or delaying recurrence of disease and prolonging survival. So the next steps are new studies rather than extending the existing trial.
Right. I suppose the problem is, although the figures look very good, as you said yourself, because it's an open-ended trial and there's no control group, it's difficult to get statistical sort of veracity to the effectiveness of the treatment.
Well, that's the normal process, I think, in drug discovery, that you would do an open trial in phase 1, early phase 2, in order to establish the maximally tolerated dose, to ensure that it's safe, and to get some initial readout about efficacy. And then that's what we've done. You create a hypothesis. We think we've created a very strong hypothesis that justifies the next step. And the next step are randomized controlled trials, which are the classic way of doing clinical trials. And that would be in a larger number of patients, and the next step.
Okay, thank you very much.
Okay.
Any other questions? Okay, then we'll move on. Just before we finish, I just wanted to highlight some of our BA upcoming events. We've got our Father's Dinner in January. If anyone's interested in coming on, we've got our Members' Day and Committee Summit in London in 25th of February. Women in Biotech in March the 8th, and Science Finance Dinner both in March in Oxford and in London. If you haven't bought your Gala Dinner ticket yet, please do so soon, because we are rapidly running out of tickets, so it will be sold out very soon. Just to conclude, I just want to say a big thank you to Richard Goodfellow and Jeff Muckle for speaking today. A really interesting webinar. The webinar will be recorded and will be sent out to everyone, so if you want to re-listen or anything like that. So thank you from the BI, and goodbye.